Only the visible light region of the solar radiation is used for photosynthesis. And as you see in the figure, visible lights begin with violet, then indigo, blue, green, yellow, and it is almost ending with orange, red, and far red. On the extreme left, there is the wavelength minimum, while as we go towards the right, wavelength increases. Wavelength increases means energy decreases. That means violet, indigo, blue region has the highest energy, while yellow, orange, red has the lowest of the energy in the visible part of the solar radiation. That means photosynthetically active radiation or PAR. These regions are utilized by the plant for photosynthesis. So, where are these pigments present? At those areas of the cell where light reaction should take place. You have studied that chloroplasts are the pigments, chloroplasts are the plastids where photosynthesis is taking place. In the figure at the bottom, you can see a chloroplast. You see, inside the chloroplast, there are the thylakoid membranes organized into grana, and a section of the thylakoid membrane is enlarged, and upon this membrane, you can see different pigment molecules arranged. And you know that all the membranes in the living system are lipid bilayer model with the proteins penetrating Singer and Nicholson type of membrane, typical membranes. A set of molecules called pigments are responsible for the absorption of energy from the light section and transforming them into chemical energy. So if the pigments are not there, light energy cannot be transformed into chemical energy. And in different plants, there are different types of pigments. For example, chlorophyll A is present in most of the plants, while phycoerythrin etc. is present in some types of seaweeds, phycocyanin also. While carotenoids, chlorophyll B, pheophytin, xanthophylls etc. are present in many types of the plants. And if we separate these pigments by means of chromatography, we can see them having different colors. What are the colors of different pigments? You see here in this slide, chlorophyll A is bluish green in color, chlorophyll B has yellowish green. So simply by looking at the color of the pigment in a chromatography paper, we can identify which are those pigments. In this figure you can see the colors which are absorbed by different pigments. Why is a leaf green in color? Because the different pigments together absorb all the other colors, while only green color is either transmitted or reflected. That's why so many different types of pigments are present in the leaf, and this is how they are advantageous. Most of the energy of the solar radiation, most of the energy of the visible part of the radiation can be absorbed. Just imagine green color or the energy from the green light also is absorbed by the pigments. Then what would have been the color of the leaves?
In your lower classes, you might have studied about the action spectrum for photosynthesis. This is that figure. You can see here an alga upon which microorganisms were distributed. They were fed into it. And T.W. Engelmann in 1882 found that these bacteria, aerobic bacteria, are accumulating in the violet blue region and also in the yellow orange red region. So from this work in 1882, Engelmann found that most of the photosynthesis is taking place in the violet, indigo, blue region and also in the yellow, orange, red region, simply because photosynthesis is releasing oxygen and these microbes are moving to those places where there are more oxygen. That is an old story. Now look at the graph given below. For example, I can show you the graph here. That graph given. This is a graph. In this graph, you can see peak. Where is the peak? Here there is one peak by around this is 400, this is 500. So around say 420, 430 region, there is a peak here, and the next peak is in the, you see, this is 600, this is 700, around 680, around like that, there is another peak here, because these are the respective wavelengths of these colors of light. So in 1882, what was done by T.W. Engelmann was actually a grand work. He did very well in that field, because his results are now proven by using modern equipment.
Here you can see the chemical structure of different pigments responsible for absorption of light. You need not draw these figures, but you should make note of one thing. All these molecules contain areas with alternate single bonds and double bonds. Look at these figures, and those areas are shaded with say violet color. These areas of the molecule with alternate single and double bonds are responsible for the absorption of light energy. So, absorption of energy is a property of the pigments. It's because of the chemical structure of the pigments.
Now we are going to see how the photosynthesizing machinery in the thylakoid membrane is organized. This is actually, can you tell me what is this? Is it a chloroplast or is it a thylakoid? You can see a cross section, not a cross section, a longitudinal section of a chloroplast which contain a lot of thylakoids. Okay, and here we are just taking a thylakoid membrane. You can see a membrane here like this. This is the membrane, lipid bilayer model. All across the membrane you can see so many things are there. You can see light is coming, something is stacked here, something called antenna, protein, carotenoid, so many things are there. Let us see what are they actually. You see, light is falling on this membranes, and these membranes are packed with so many different types of pigment molecules. You can see here the term pigment. Remember, chlorophyll is just one type of pigment. So many different types of pigments are there. For example, carotenoids, xanthophylls, etc. And chlorophylls also are there. You can see here carotenoids. So the pigments are arranged on the surface of this membrane. Which membrane? Thylakoid membrane. So light is falling upon the pigments in the form of photons, and that energy is somehow reaching a place called reaction center. Actually, what's the reaction center? It's a set of chlorophyll molecules where the first reaction is taking place. Here, the energy of a photon is used for the separation of charges. It is initiating electron flow. What's a photon? You know that photons are actually making up the solar radiation. And not only these green pigments, but many other different pigments are there. We say they are all antenna chlorophylls. Antenna chlorophylls are there. Other antenna chlorophyll molecules are there. And another category is their accessory pigments. Carotenoids and different other pigments are there. They form the accessory pigments. So these antenna chlorophylls, antenna molecules and accessory pigments all together absorb light energy. Transferring is between molecules until it reaches the reaction center. So the energy being absorbed by one molecule is given to the next one, from there to the next one, from there to the next one, until it is reaching the reaction center. So that is what is taking place here.
What are the components of the molecular machinery required for light reactions? Photosystem is the name given to the functional unit of light reaction. So a photosystem consists of all the molecules required for light reaction to take place. It's a basic unit. Basically, a photosystem consists of a reaction center surrounded by a number of light harvesting complexes, LHC. What's the reaction center and what is an LHC? A light harvesting complex is an assembly of pigment molecules along with associated proteins. There are different types of proteins associated with it. While a reaction center is a protein complex which consists of two special chlorophyll A molecules and a primary electron acceptor. Remember, there are two types of photosystems in plants, photosystem 1 and photosystem 2. How do they differ? We will see in the following slides. In this slide, you can see the two photosystems, photosystem 1 and photosystem 2. They are named 1 and 2 simply in the order of discovery. Photosystem 1 is the one which is discovered first. Then only photosystem 2 was discovered. How do they differ? Basically, the difference is in the wavelength at which their reaction centers are absorbing. In photosystem 1, the reaction center absorbs light at 700 nanometer, while in reaction center 2 or in photosystem 2, the reaction center is absorbing light at 680 nanometer. That's a basic difference. And you know that photosystem 2 is utilizing water. It is broken into electrons and protons and oxygen. Oxygen is released. These things are not taking place with the photosystem 2. At the same time, both are absorbing light energy and electrons are excited and light reaction is taking place. And you will see later that there are two types of flow of electrons in this process. Photosystem 2 is taking part in the non-cyclic flow of electrons, while photosystem 1 is taking part in both the cyclic and non-cyclic flow of electrons.
Now let us see on a thylakoid membrane where the photosystems are located. Actually, there are separate areas for the photosystem 1 and also for the photosystem 2 in the thylakoid membranes. Before that, you should be familiar with two terms. One of the term is oppressed region. Oppressed regions are actually the regions of the membrane which are stacked. Stacked means you can see layers like this, this is layers you can see like this. Okay, so such a region where the membrane is stacked one upon the other, that is what is called oppressed region. Here this is the oppressed region, and here in these areas you can see it is unstacked. Unstacked region means non-oppressed region. So two types of areas are there on the thylakoids. Now this LHC, light harvesting complex 2 and ATP synthase are located in the oppressed regions of the thylakoid. You can see here light harvesting complex 2 is there, ATP synthase is there, photosystem 2 is there. All those are located in the oppressed regions of the thylakoid, also called granule lamellae, in which several membranes are in contact and in regions that are not oppressed and have already access to ADP and NADP systems in the stroma. For what purpose? For this photosystem 2's functioning. Already we found that photosystem 2 is involving in non-cyclic photophosphorylation activities. What about photosystem 2? It is exclusively present in the oppressed regions, and photosystem 1 is almost exclusively in the non-oppressed region. So, here is the oppressed region, stacked region. You can see here the photosystem 2, light harvesting complex 2, etc., etc. While inside you cannot see any photosystem 1. Here, this is photosystem 1. This is photosystem 1. You see, this is photosystem. See, the photosystem 1 is present exclusively in the non-stacked region, non-oppressed region, in the exposed region. Okay. So, in the stacked regions, photosystem 2 and other their related components can be seen, while in the non-oppressed region, in the unstacked region, you can see photosystem 1. How is the energy being transferred? The excitation energy is being trapped by the photosynthetic reaction center. You can see here, these are the antenna pigment molecules. Energy is falling upon it, E is equal to h mu, and that's this energy level is actually excited. These molecules are now excited. These excited molecules are transferring their energy to the nearby molecule like that. Ultimately, they are reaching the reaction center chlorophyll. Why? This light energy that has been passed among pigment molecules by excitation transfer is trapped by the reaction center chlorophyll here, because this reaction center chlorophyll is its lowest excited state. In that state, it has a lower energy than those of the antenna pigment molecules. You see, the pigment molecules are at a higher energy level when they are excited, and when excited, reaction center chlorophyll is at a higher energy level than when it is at the ground level. But in the excited state, the reaction center chlorophyll is at a lower energy level compared with the energy level of the excited antenna pigment molecules. You can just imagine it like this: water is freely flowing from a higher level to a lower level. Like that, energy can be easily captured by the reaction center chlorophyll, which is at a lower energy level compared with the excited pigment molecules.
How is the photosystem working? Light is falling on the pigments in the form of photons. They are absorbed either by the reaction center or by the accessory pigments. If the photons are absorbed by the reaction center, then the reaction center is emitting electrons, because by absorbing energy, electrons are excited and the electrons are thrown away from that molecule, which is absorbed by a primary acceptor, and the electron flow begins. On the other hand, those photons which are falling on the other accessory pigments, other chlorophylls are there and carotenoids, xanthophylls, etc. are there. They are transferring their energy through a series of pigments of the antenna complex. Ultimately, they also reach the reaction center chlorophyll A. And in response, chlorophyll A is emitting electrons. That's why these chlorophyll A molecules are said to be the reaction center.
Which are the stages of light reaction? There are 4 stages which you see here in this slide. First one is a primary photo event, then there is a charge separation, then there is electron transport, and fourth, there is chemiosmosis. What is the primary photo event? A photon of light is captured by the pigment. The result of this primary photo event is the excitation of an electron within the pigment molecule. In the previous slide, we found this one. Second is the charge separation. This excitation energy is transferred to a specialized chlorophyll pigment. It is called reaction center. We found earlier this term also. So, reaction center is the chlorophyll pigment molecules which is accepting the excitation energy. And this reaction center responds by transferring an energetic electron to an acceptor molecule, thus initiating electron transport. In some of the pathways, thus this acceptor molecule is called pheophytin. I will try to write it here. It is called a pheophytin. That term is not mentioned here. Pheo, P-H-E-O, pheophytin, P-H-Y-T-I-N, pheophytin. It is the primary acceptor molecule, but that name is not mentioned in this one. It is there in the next slides. Now there is the electron transport. The excited electron is shuttled along a series of electron carrier molecules, just as in the case of mitochondrion. These electron carrier molecules embedded within the photosynthetic membrane. Several of them react by transporting protons across the membrane. The best term is several of them responds. That's actually a better term. By transporting protons across the membrane, which means they are acting as proton pumps. This generates a gradient of protons, proton concentration gradient. Its arrival at the pump induces the transport of a proton across the membrane. This electron is then passed to an acceptor. Then you may have a doubt, from where is the electron taken for this pumping? In which direction is the proton accumulating, or are they being pumped? We can see it in the coming slides. And the last stage is chemiosmosis. You might have heard of chemiosmotic hypothesis as proposed by Peter Mitchell. The protons that accumulate on one side of the membrane now flow back across the membrane through a specific protein complex where chemiosmotic synthesis of ATP takes place. This is just as we found in the case of a mitochondrion. Actually, in both the cases, basic principle is almost the same. We will see how this is taking place in the next slides.
As you are all aware, photosynthesis actually consists of two phases. The first phase depends upon energy from the light or a series of light dependent reactions, while the second phase independent upon energy from the light directly, or we say the second phase called dark reactions is making use of the products of light reaction. And in this figure we can see that water is utilized, oxygens and protons are released, ATP is released, and NADPH plus H plus also is released. So these are the products of the reaction. Of these, the electrons released from water are transported along the electron transport system. The oxygen gas diffuses into the atmosphere. The protons and electrons are utilized for the formation of NADPH plus H plus. So ultimately, ATP and NADPH plus H plus are the products of the light reaction to be utilized for the dark reaction. Let us see these pathways in detail in the following slides.
Okay, now let us see in which direction are the protons accumulating from this diagram. Earlier in a slide we found that the electrons are flowing through the electron transport system along the thylakoid membrane, and it is resulting in the pumping of protons. Can you see here? In this space so many protons are there. Which is that space? It is the thylakoid lumen. while outside there are so little protons. It is because protons are actively being pumped into the thylakoid lumen when the electrons are flowing along the electron transporter molecules present along the thylakoid membrane. And at the time of ATP synthesis, these protons are diffusing back into the thylakoid, back into the stroma from the thylakoid membrane, or from the, better we say, from the thylakoid space, thylakoid lumen, across the thylakoid membrane. Of course, you have seen that picture here that ATP synthase molecule is there. These protons are diffusing through the F0 base of the ATP synthase molecule. And this is ultimately resulting in the synthesis and the release of ATP. You have seen that just earlier. We are also going to see the mechanism of chemiosmosis. Now, you need to keep this diagram in your mind, that when the chloroplast is actively synthesizing, the proton concentration is too much in the thylakoid lumen and too little in the outside space stroma, so that a concentration gradient of the protons exists across the thylakoid membrane. Because of this gradient, protons are forced to diffuse across that membrane through the F0 F1 particle, through the ATP synthase particle, from the thylakoid lumen towards the stroma. Let us see a model of the thylakoid membrane. Of course, you have seen this in the earlier slides also, but this is from another textbook. From the books I gave you, you can easily find out from which textbook is this figure taken. This will help you to identify the suitable books and to prepare for your examination. Okay. As you see here there is a chloroplast. You can see the outer membrane of the chloroplast, inner membrane of the chloroplast, thylakoid, stroma, etc. And the thylakoid membrane is being expanded here. You see, according to this diagram, water is being split, electrons are flowing along this electron transport system, protons are released into this space, which is this space. This is the thylakoid lumen. Oxygen also is released here, and we are not concerned about oxygen here, because in the process of photosynthesis, oxygen is a byproduct. So let us just forget it. Let us go after electrons and protons. And when the electrons are flowing through these carriers, you found earlier also that some of these are acting as proton pumps. So protons are being pumped into this thylakoid lumen. So thylakoid lumen is getting protons from two sources. Source 1 by the photolysis of water, by the breakdown of water. Source 2 being pumped by some of the electron carriers along the membrane. You can see it here. And the proton is being lost from the thylakoid lumen towards the stromal side of the chloroplast. Look here, this is the stroma. Outside the membrane is the stroma. The protons are lost from the thylakoid lumen to the stroma. Look here, only through one channel, that is the proton translocating ATP synthase. You know that when the protons are being transported like that, when they are diffused like that, ATP is being synthesized and released. Look here, these are the various transporter complexes. One is the PS2 complex. PS2 complex is there. Electrons are being excited. They are being absorbed by P680, pigment 680. It is being from P680, the electrons are accepted by pheophytin. I told you earlier, pheophytin is the first acceptor. Then quinones are there. Quinone becomes quinol. Like that, it is being transported. Here, actually, quinone becomes a quinol. and from here it is coming to cytochrome B6F complex, etc. That is very clear here. You found in the previous slide also. What is important here is that in this diagram both the photosystems are involved. See here, photosystem 2 is there and photosystem 1 is there. And since two of them are involved, the electrons are flowing in a one-way direction. The electrons which are starting from here are not coming back. So this is a non-cyclic flow of electrons. Point 1. This is a non-cyclic flow of electrons. And this non-cyclic flow of electrons is being triggered by light. It is being triggered by light. Because when light is falling upon this pigment system, energy is being captured. Another point is that when the electrons are flowing like this, protons are accumulating, creating a proton gradient, and these protons are flowing back through this ATP synthase, and ATP is being made. Synthesis of ATP is phosphorylation. Actually, this is photophosphorylation. Why do we call it photophosphorylation? Because synthesis of ATP being triggered by light. That is what is called. Ultimately, it is being triggered by light. So we call it photophosphorylation. And the electrons are flowing in a non-cyclic manner. Therefore, we call it non-cyclic photophosphorylation. Don't worry, we will see one more slide about this one. And what happens to the electrons and protons and oxygen? Of course, you know, oxygen is simply diffusing the atmosphere, so don't worry about it. And protons are being accumulated in this thylakoid lumen, and they just pass out through this transporter protein. Electrons are being transported, look here, from the PS1 complex. They are going to iron sulfur, means ferredoxin. From there, they are going on to ferredoxin NADP plus reductase. This enzyme is located on the thylakoid membrane. Ferredoxin NADP plus reductase. What is it doing? It is accepting NADP plus. It is accepting electrons. It is accepting protons. So this ferredoxin NADP plus reductase is accepting three types of species. One is NADP plus, the other one is protons, and the third one is electrons. Ultimately synthesizing NADPH plus H plus molecule. Ultimately synthesizing NADPH plus molecules. You know that this light reaction is consuming light, it is consuming water, and it is producing ATP and NADPH plus H plus. And these NADPH plus H plus and ATP are being, or they are going to be used in the dark reaction. So make a note of it and keep it in your mind. A schematic representation of non-cyclic photophosphorylation. What you see here is actually a graph. What is there on the x-axis and y-axis? On the vertical axis, you can see oxidation reduction potential in volts, relative energy level. You can see it here. Okay, and don't worry about the horizontal axis. It is just showing the sequence of the reaction. See here, solar radiation is being absorbed by the pigment molecules. They are being transferred from molecule to molecule, ultimately reaching reaction center. In the reaction center, electron is excited and it is absorbed by a primary acceptor. Remember, here the species are arranged in the order of their oxidation reduction potential. You should keep in mind that all these components are present along the same membrane. They are not separated in space, they are present all along the same membrane. Here in this diagram, you see them in different places because they have different oxidation reduction potential. Keep it in your mind. So here there is the reaction center. Here is the primary electron acceptor. So from the diagram it is very clear that the primary electron acceptor is at a higher oxidation reduction potential compared with the reaction center. That's all. And it is then given to plastoquinone at a lower voltage, I mean at a lower potential, then to cytochrome complex, then to plastocyanin, etc. In some of these stages, you know, ADP is being used for synthesis of ATP, and it is reaching photosystem 1. Remember, in non-cyclic pathway, both the photosystems are participating. Look here, which one is at a higher oxidation reduction potential, photosystem 2 or photosystem 1? Just make a comparison. And electron is reaching only up to photosystem 1. From there, it is excited once again by new photons absorbed from the solar radiation. This time it is again absorbed by another primary electron acceptor, and it is given through a series of acceptors and donors. Ultimately it is reaching ferredoxin, and here we have the associated with this ferredoxin we have the NADPH synthase as well. It is accepting NADP plus and protons and also electrons, synthesizing NADPH plus H plus. So keep it in your mind. Let us come to cyclic photophosphorylation. In another slide earlier I told you that there are two types of photophosphorylations. This is cyclic. Why do you call it cyclic? Because the electrons which are starting from P700 are moving through some acceptors and carriers, donors, etc., and they are coming back to the same system. So the electrons are moving in a cyclic pathway, and in some of the stages there are sufficient proton pumps so that ATP is being produced. But remember, here water is not split, protons are not released, NADP H plus is not produced, oxygen is not released, and it is involving only photosystem 1 P700. It does not involve photosystem 2, and you can see the components here very clearly: primary acceptor, ferredoxin, cytochrome complex, plastocyanin, etc., and the electron is just reaching back. This electron is excited to the higher energy level because of the energy being absorbed from the light. So it's better if you make a comparison between cyclic photophosphorylation and non-cyclic photophosphorylation.